right, so we're going to talk about DNA replication, which is exactly what it sounds like. It is the process of DNA making more DNA. So if DNA needs to replicate itself, it's going to make more DNA. Why does DNA need to make more DNA? No, that's not a rhetorical question. I'm actually asking you. Why does DNA need to make more DNA? Hopefully what you said was that when a cell is going to duplicate, so when one cell is going to become two cells, it has to make a second copy of the DNA in order to pass on to the daughter cells. You learned that back when we talked about mitosis and the cell cycle. So when specifically is replication going to happen? When in the cell cycle? Yeah, I'm asking you again. Come on. Okay, so hopefully your answer was the S phase of interphase. Remember when we talked about the cell cycle, I said during the S phase of interphase, the DNA replicates. And I said, don't worry about that right now. You'll learn more about it later. Haha, it's later. It's time to learn about DNA replication. So during the S phase of interphase, the DNA replicates so that then the cell can move on and go to G2 and then go through mitosis and cytokinesis. So we're going to start with helicase. Now you can see helicase, if you look at that word, it ends in ace. So ace tells you you've got an enzyme, and hela, like helix, it tells you that what helicase is going to do is it's going to unwind and unzip the helix. So the helicase is going to come in and it's going to split apart or unzip our helix. Now you see all these guys floating around. It looks like this dude is like popping those off of there. These are called free nucleotides. Notice nothing is actually popping off of here. So all the helicase is doing is coming in and opening up this DNA strand. So it's going to come and open up this DNA strand until we have what's called a fork, a replication fork. And that's because it is a fork, like a fork in the road. Two roads diverged on a snowy night. Oh, wait, that's a whole different subject. Okay, so then there's DNA polymerase. Again, ACE, that's another enzyme. Polymerase, it's an enzyme that's going to make a polymer, and it's DNA polymerase. So what polymer is it going to make? It's going to make DNA. So if you forget what it does, it's right in the name. It's an enzyme that builds the polymer of DNA. So DNA polymerase is going to come in, and it's going to attach individual nucleotides to the strands that are hanging free. So polymerase comes in, and it says, hey, I see you've got a cytosine here. I'd like to give you a guanine. I see you've got an adenine here. Here's a thymine. Go ahead and attach. Here's a guanine. Have a cytosine. There's another DNA polymerase down here, and it's saying, I see you've got an adenine. You would like to have a thymine, wouldn't you? I see you've got a cytosine. Go ahead and take a guanine. And the polymerase is going to go on down the line, attaching free nucleotides onto the existing template strands. Well, it's not quite that easy. It's a little bit more complicated. You already know that DNA is directional. Okay, so remember we said that DNA is built in two directions at the same time. So we've got sort of, if you think about our pentagons for our deoxyriboses here, notice that the top, that oxygen at the top is at the top here, and that oxygen is at the bottom down here. So we've got everything is facing opposite directions. So now we're going to name those ends, we're going to name those directions. So this end of this molecule, this side of the molecule, this is what we're going to call the 5' end, that little apostrophe, we say that as prime, so it's the 5' end. And down here we have the 3' end. So then logically the other side, where this one has a 5' end, this strand is going to have a 3' end. And where this strand has a 3' end, this one's going to have a 5' end. So it's important to know which direction each strand is running because there's an important rule. Replication is going to add in the 5' to 3' direction of the new strand. Now, I do not expect for you to remember molecularly what it looks like at the 5' end and what it looks like at the 3' end. I will tell you where 5' is and where 3' is on a given strand. You need to remember that you need to add 5' to 3' of the new strand. Okay, so when our new strand comes in, it's going to start down here because the 3' end of the old strand is where the 5' end of the new strand is going to be. And then replication is going to go in this direction. until we get to where we've got a full strand that completely matches. That new strand is called the complementary strand. So you have the template strand, this one that was pre-existing, and then you have the complementary strand here that is not pre-existing. It's the new one. But of your two strands, only one of them is actually going to be open and out there enough for the building to continue uninterrupted. And I'll explain that in a second. But that one's called the leading strand. So the leading strand is the one that can continue uninterrupted. So if you look at the way this template strand has been split open, here's the 3' end, which means this new strand is going to start 5'. And so it can continue in this direction. Remember that helicase is right here. And helicase is going to keep moving this way and opening up this existing DNA strand. Okay? So as helicase moves down this way, this polymerase can just follow the helicase and build that strand. So there's no problem. So this is the leading strand because it can just continue on uninterrupted. But the other template strand has the 5' end hanging out free. So if we were to attach a new nucleotide right here, we would be attaching a new nucleotide in the 3' at the 3' end of our new strand. And we can't add a free nucleotide to the 3' end of our new strand. We have to start at the 5' end of our new strand. So the 5' end of our new strand is going to be down here. Okay? And we build 5' to 3', which means this new strand is going to start here and build this way. And so if this strand polymerase starts here and builds this way, while it was doing that building, helicase came and opened up more of them. So what you're going to have happening is that your polymerase is going to start here and then go this way. And then it's going to have to move back to where the new fork is and then build, and then move back to where the new fork is and then build, and then move back to where the new fork is and build, and move back and build, and move back and build, and move back and build. And so you're going to end up with all of these little pieces that were individually built. Now, in reality, you're not going to have one polymerase that's zipping back and forth like that. You're going to have multiple polymerases that are all attaching on as they can as a new fork is opened up. So these mini fragments are called Okazaki fragments. I love that word. Okazaki fragments. So you've got all of these little pieces here. So a third enzyme is going to come in here, and it's called DNA ligase. If you've ever heard of a ligature is a tie that binds something, that holds something together. So ligase is an enzyme that attaches pieces of DNA together. So DNA ligase is going to connect the Okazaki fragments to each other, and it's going to create one complementary strand. So here is a chunk of DNA that got created. Here's another Okazaki fragment. So we've got Okazaki fragment, and DNA ligase is connecting them together. And then we have another Okazaki fragment that's just finishing up right now. And when this polymerase releases and goes back to the replication fork, ligase can come in and attach these guys. And so you're going to end up with one whole new complementary strand. But that one's a little pokier. It takes more time to make that happen. So it is called the lagging strand. It's the lagging strand because it lags behind. Replication is complete when you've got two daughter strands left. So here is your original strand, and here are your daughter strands. Each daughter strand consists of one original strand, the template strand, and one new complementary strand. So notice that these guys are both blue, okay? So here you have one blue template strand, and then one new yellow complementary strand. Again, here you've got the other old blue strand, template strand, and the other new complementary yellow strand. We call this semi-conservative replication because we aren't 100% conserving things. If we were conserving things, what we would have is we would have one entirely blue strand and one entirely yellow strand, and we would have one that's all old and has been conserved or saved, and one that is completely new. We don't want that because if we have that happen, then you're going to have this guy that after many, many copies is going to get old, and you're going to have greater chances of mutations going on. Pieces are going to fall off, and we're going to be continually replicating places where, things are just, it's decaying, it's not doing well. Okay. So as a quick overview, we have our original DNA, which is 2 strands moving in opposite directions to each other. We're going to unzip our DNA, add complementary base pairs in the 5' to 3' direction of the new strand, and we're going to have our one leading strand and our lagging strand that consists of Okazaki fragments. Okay, remember it was helicase that unzipped the DNA, polymerase that built the DNA, built it, and then ligase is going to come connect those complementary base pairs until we end up with our new daughter strands that have one old template piece of DNA and one new complementary strand of DNA. Then over here we have one new complementary strand and one old template strand, and so we call it semi-conservative replication. And that is all.